So here we have chapter 13, which is on viruses. I'm going to cover prions separately. So this is just going to be on viruses. All right, so often people say the viruses are not living, and that has to do with the fact that all they really are is nucleic acid covered with protein. The nucleic acid could be DNA or RNA, but the genes on, let's say, the DNA code for how to do infection and then how to make copies of themselves once they've taken over a cell. But they have to have the cell. They have to have the cell's cellular machinery. They have to have the cell's DNA. They have to have the cell's building blocks. So they are the ultimate parasite. And then very often the strategy is to make as many copies as they possibly can in a short period of time, but then they've used up all the resources and the cell dies, and then the new viruses break open the cell and are released to infect even more cells. So it says viruses contain DNA or RNA and a protein code. Now, the protein code is to protect it. Its blueprints, if they're destroyed, that's it. So they very often have one or two types of coating around it so that they're protected until they meet a host cell that they can parasitize. And so that's why some are also enclosed by an envelope. Most viruses infect only specific types of cells in one host. So there's a virus that causes leukemia in cats, and very often kittens are inoculated against the feline leukemia virus. But if your cat does develop feline leukemia, the cat cannot give you leukemia. Very often they are that very specific, like HIV is human immunodeficiency virus, and it cannot infect your dog. So that's what they mean by very specific types of cells. And that could be between species. It also could be that the influenza virus is going to infect one type of cell in the lung. It's not going to infect cells in the pancreas. So that's the other type of specificity. And the host range is determined by specific host attachment sites and cellular factors, which we will go into. Now, they're very small. Here's a human red blood cell, and here's an E. coli bacterium. So the E. coli bacterium is about 10 times smaller than the human red blood cell. Then take the very small bacteria, like E. coli, and look at the sizes of the viruses. You know, here you have a bacteriophage that's probably 100 times smaller than the E. coli. And even the ones that are pretty big, like Ebola, it's only maybe 10 times smaller than the E. coli, but smaller it is. Viruses are very small, and then you have the bacteria, and then you have mammalian cells. All right, so here's the darling of viruses. It's been studied the most. It infects E. coli, and because it has a very specific range, people didn't mind working with it because it wasn't going to harm them. And E. coli is very easy to get. Everybody has it in their large intestine, and so forth and so on. So it's called the T bacterial viruses, and this one is called T4. And for some reason, people decided to call bacterial viruses different than animal viruses. Animal viruses are called viruses, like HIV is a human immunodeficient virus. For some reason, all bacterial viruses are called bacteriophage, because phage comes from the Latin, which loosely translates into virus. Don't ask me. It's just one of those things that the student gets to learn the name virus and the name bacteriophage, and they mean the same thing. So this is the darling that's been studied so extensively, and it has a head, which is called a capsid, and it has a sheath, and then it also has these tail fibers. Sometimes it's called the head, the tail, and the tail fibers, and I like that because it makes more sense to me. Inside the head is the DNA. T4 is a DNA virus, so the blueprints for making T4 viruses or T4 bacteriophages are located in the head, right up here. And all of this is protein, the outer protein coat. Then when you have the tail, that's going to operate like a hypodermic needle. And you're going to say hypodermic needle? You're kidding me. Very often Mother Nature has figured out how to make things like hypodermic needles long before we did, and this is an example. You're going to find that the tail is actually made of contractile proteins. And then you have the tail fibers. And although you can't see it, yes, you can. You see these little pins at the bottom? They actually act like a needle because they're going to be able to pierce through the cell wall and cell membrane of the E. coli and then inject the DNA. So these pins, the tail fibers will kind of stabilize it on top of the E. coli, and the pins will pierce it as the tail contracts and pushes, just like a hypodermic needle, all the contents of the viral DNA into the E. coli. And then once this outer shell of protein has inoculated or injected the bacterial DNA into the E. coli, it just falls off. It's empty and it just falls off. So here we go, T4. And now we're going to do a little bit of viral taxonomy. We've already talked about the fact that bacterial viruses are called bacteriophages. I'm not going to test you on this at all, but if you're just dying to know, a family of viruses end in Viridae. The genus name of a virus ends in virus. There are different viral species, and that's going to be very much viral specific. So bacterial viruses are named a little bit differently than animal viruses. Now, subspecies are designated by a number, and this tends not to be that important unless there are major epidemics caused by a subspecies. So E. coli 157, there have been numerous breakouts back in the 20th century of this particular type of E. coli, and it's quite virulent and dangerous, and it can cause bloody diarrhea and things of that nature. So E. coli usually is a very friendly bacteria in your gut, but this particular subspecies has mutated into a pathogenic form. So people would say, oh my God, what you have is E. coli, 157, and that is the reason why your stools are bloody and you're so sick. So subspecies tend to give you more information about the organism that you're studying. Okay, so now this is also showing you some types of viruses. And here you have herpes, viridae, herpes virus, human herpes virus 1, HHV2, HHV3, and so forth and so on. And of course, one of the most famous is HIV. Okay, so what's the multiplication? Since the bacteriophage of T4 has been studied most extensively, and that the strategy of viruses, whether they're bacteria or human or animal, is exactly the same. The idea is that you're going to take over the cell, try to divide as quickly as possible, and then the cell lyses and dies, and the new viruses are released. So it's called the lytic cycle because the host cell is going to die and lyse. So the first thing that's going to happen is the virus is going to attach. And this can be very specific or very general. In this case, the bacteriophage of T4 attaches by the tail fibers, kind of like landing gear, to the host E. coli. Then you're going to have penetration. So this is the injection of the viral DNA into the cell. So it says there is a enzyme that can open up the cell wall, and the tail sheath or tail contracts to force the DNA into the cell. The next is going to be just what bacteria, or what happened to bacteria, not happened to us. Remember, they only have one chromosome. So what the virus that attacks the E. coli, or a virus that attacks bacteria, does next is to destroy the host DNA, to destroy the E. coli circular chromosome. And of course it has a way of not destroying its own DNA, and that's another discussion, but the virus can very specifically just destroy the host and not its own, because it wants to tell the cell what to do. It doesn't want to compete. So the first thing that it's going to do once it takes over the cell is to do biosynthesis. So the instructions that are encoded in that bacteriophage or virus is going to now take over and instruct the cell to synthesize as many of its parts as it possibly can. So it produces more DNA, and it produces all kinds of proteins, like the head proteins, the tail fiber proteins. But it doesn't like a car assembly line. It doesn't doesn't build one and goes on to the next and go on to the next. Again, viruses realize that if you make the parts list first and assemble them second, you can do it quicker. So the next is assembly. It's called maturation or assembly. There is quite literally assembly line, and everything is now put together. And then once you've made all of these new viruses, you're going to stop doing it when you run out of supplies. And that then signals the release of the new viruses. And there is an enzyme that can break open the cell wall of the bacteria called lysozyme, and all hundreds of new viruses are released, and they just diffuse and attach themselves to new bacteria, and so it goes. So that is a lytic cycle. So this is showing the lytic cycle. Here you have the little bacteriophage T4. It lands with the tail fibers. It injects the DNA. It then takes over after it destroys the host chromosome and synthesizes the parts. You assemble the parts, and then you break open the cell and release them. Big difference between bacteria and our cells is that we have 46 chromosomes. So it's going to be like nearly impossible for a virus, a tiny little virus, to destroy 46 chromosomes. So what they do instead is they have another strategy, and that is to inactivate our chromosomes, so that the only piece of DNA that's going to be able to be transcribed and translated is the viral DNA. Okay, so that's a big difference. Another thing that can go on with animal viruses, mammalian viruses, viruses that infect humans, and viruses that affect bacteria is you can have two types. You can have a bacteria that, a virus that does a lytic cycle, so that it takes over, makes copies, it destroys the cell. In some cases, you have what is called a lysogenic cycle or a latent cycle. Not all viruses can do this. Some viruses can. They become very famous. Famous ones are herpes. Herpesviruses can cause anything from chickenpox to shingles to cold sores. And the herpes virus is a family of viruses that can do either a lytic cycle or a lysogenic cycle. The lytic cycle all viruses can do. So all viruses can in fact take over, make copies, kill the cell, release it. Lysogenic or latent means to hide, and it's going to hide away in the cell. And when you get chickenpox when you're 3, it's able to hide inside cells for decades. And the herpes the herpes that cause chickenpox at 3 can cause shingles at 63. And that virus has been copied faithfully for all those decades. So lysogenic cycles mean that now prophage is another name for virus. So the viral DNA can incorporate into the host chromosome. And the trade-off is this. The trade-off of a lysogenic cycle of a bacteria is that, yes, it's going to incorporate, but the incorporation means that the virus, when the cell divides, will be faithfully copied. That's the reason why at 63 you can get shingles from that herpes virus that gave you chickenpox when you were a kid. But when it does that, it cannot cause disease. So when you're 63 and you get run down, or you might be on chemotherapy, which weakens the immune response, then somehow that herpes virus understands that the immune system is compromised, and it will be able to pop out of the chromosome and then start a lytic cycle that ends up causing a totally different disease like shingles. But shingles means that that virus is in a lytic cycle. Then you can, treat the person with drugs that will inhibit viral replication, and then once the person's immune system is strong enough, then they've recovered. But you never know if you have destroyed all of that virus, or that some of that virus has gone into hiding once again. So that's kind of the up and down side of a lysogenic cycle. So here's a example of a lysogenic cycle. So this is a virus, right, that it's going to inject its DNA. But instead of destroying the bacterial chromosome, it's going to, in number 3, so it's going to do in number 5, sorry about that, it's going to take that little red bacterial virus and it's going to incorporate in to the host chromosome of the E. coli. So when it does that, it's not going to cause disease, and it's not going to take over a cell, and it's not going to destroy the cell. Instead, the cell is going to grow and multiply and copy that viral DNA over and over and over and over again. And then what can happen, and it's called either the virus or prophage. They mean the same thing. And then something can happen to the cell. And in humans, very often, it's something that is going to compromise the immune system, because the immune system is there to help defend the body against viral infections. But if the virus knows that the immune system is compromised, then it can pop out and start a lytic cycle. And again, you can treat it, and you're starting to destroy the virus, but some of the virus is going to go into hiding, and it will then be the type of thing that the person's going to now seemingly recover, but you don't have any guarantee that you've gotten rid of it. So cold sores, once you've got a cold sore, it will erupt, let's say, when you have a cold and your immune system is compromised, and then you recover from the cold and your immune system is fine, and then what happens is it goes back into hiding. And then when the person, perhaps maybe a year later, 2 years later, same thing happens, they've got themselves another cold sore. Now, multiplication of animal viruses. This is the last slide of this particular conversation. The animal viruses, let me go back, sorry. The animal viruses are very similar in their strategy, but they've obviously made some compromises, because you're talking about an animal cell. We're talking about a lot of chromosomes. We're talking about a cell that's more complicated, that has organelles, etc., etc. So in general, whether you have a DNA or RNA animal virus attack your Homo sapiens cells, you do have attachment, and they're going to attach to the cell membrane of a human cell. They're going to penetrate usually by fusing. And when we do the next chapter on HIV, this will become clearer. It's going to take off its protein coat. It's going to synthesize its products. It's going to assemble or mature. The release very often is by budding, but it also can rupture. And we'll talk about the differences. And I'll just mention it right now. If you're talking about the influenza virus, it wants to hit quickly. It wants to replicate as fast as it can, and it does destroy the cell. But it's a cell that can replicate and replace itself. And it's trying to cause symptoms that makes the person cough and sneeze and create an aerosol that will infect other people. And so it's the idea of hit it fast, make a lot of copy, kill the cell, spread to a neighboring person, and start it all over again. Budding tends to happen with viruses that are sexually transmitted. And I'm going to pick on that because what the virus wants to do is not kill the cell, because it wants to slowly replicate by budding. But when it buds out of a cell like HIV does, it doesn't kill the cell. It just takes a little bit of cell membrane and it buds off. The idea of sexually transmitted diseases, whether it's bacteria or viruses, is they want to keep the person healthy. They don't want the person to appear sick. They want to, however, slowly divide so the person is infectious, even though the person feels fine. Why? Because it's sexual reproduction, and people, this is going to sound silly, they don't want to have sex with people who are really sick. If the person seems healthy, wealthy, and wise, then hey. But if the person looks like they're on death's door, that's going to be different. So the strategy of a sexually transmitted disease is going to be vastly different than the strategy of a disease that causes the flu. So that's the reason why it's complicated, and budding is going to be more of a strategy of let's keep this individual healthy and until you can't stave it off anymore, versus I want to get in quick and do rapid reproduction, kill those cells, spread, and that's how I'm going to cause disease. That's the end. So next time you listen to me, it's going to be about HIV and prions.